So lysogeny means that once DNA is integrated into the bacterial chromosome from a bacterial phage, that the cell survives. It's going to live and be able to replicate that DNA and pass it on to its offspring. There is a cycle that's referred to as the lytic cycle, where where you will see essentially the rupturing of the host cell. Because what happens is that viral DNA is integrated into the bacterial DNA, and then the viral DNA starts to break apart the bacterial DNA, and it starts to develop these phage heads. And then those phage heads overtake the cell and cause the cell to lyse or to rupture. A phage is known as a prophage when it's early, when all that remains of it is its DNA. So phage typically has these proteins that surround it, or that surround the DNA inside of it. But once those proteins and that outer capsule is kind of lost, then you just simply have a prophage, which is just the DNA of the virus that's going to integrate into the bacterial cell. Now, the bacterial cell that ends up taking up or integrating the viral DNA into it is going to be referred to as a lysogenic cell. And then as the bacterial cell starts to display the starts to express the viral genes, then it's going to be referred to as a lysogenic conversion. So that means that the bacterial cell is taking on and expressing any of those genes that were encoded by the viral DNA that was integrated into it. So here's a nice diagram of what happens in the lytic cycle and lysogenic cycle, which hopefully will help you understand it instead of looking at all that text on the previous slide. So for the lytic cycle, remember what happens, and this is kind of a continuous process, but we'll start here. We have the bacteriophage, which you can see here, and the bacteriophage, like I said, it looks like a syringe pretty much. The bacteriophage has this red strand of genetic material, which can be DNA or RNA. In this instance, it'll be DNA. And then the DNA of the bacteriophage is released into the bacterial cell. The bacterial cell's DNA will integrate the viral DNA into its chromosome. And then this viral DNA is going to start to break apart this chromosome in the lytic cycle. So you see how the chromosome of the bacterial cell has been split apart. So now that the chromosome has been split apart, these viral pieces of DNA start to package themselves into the components that are left over by the bacterial cell's DNA to formulate what we call phage heads. And so the external structures of this bacterial phage are starting to be developed, and the DNA of the virus is starting to be packaged inside of those bacterial phages. So once these bacterial phages start to assemble, so they and start to mature, then they're going to push themselves out of the cell by causing the cell to rupture through inundation. And so these bacteriophages are going to leave the cell and find other host cells to inject themselves into. Now, when a bacteriophage lands on a cell and it releases its viral DNA and the viral DNA is integrated, there's two pathways that could be followed, either the lytic cycle, which we just talked about, or the lysogenic cycle, in which now you have essentially a lysogenic cell, which is going to simply replicate the viral DNA. And as that viral DNA is replicated, then the cell will undergo the process of binary fission, where the cell is going to be splitting apart. And now you have two cells that have the viral DNA that's been integrated into them. So there's no harm done to the cell in this instance, so it's referred to as the lysogenic cycle. So this is what we're talking about when we talk about lysogenic conversion. Now, we're going to talk about the process of transduction, which is essentially the same thing as the lytic cycle. Now, in the process of transduction, you're going to have a bacterial phage. It's going to integrate its genetic material into a recipient cell. that genetic material will break apart the bacterial chromosome, and then those phage heads will be developed. And once those phage heads are developed, they'll rupture the cell and go and attack a new cell. And then once that new cell has the bacteriophage attaches to the new cell, then it's going to unleash its genetic material and integrate that genetic material into the new cell. So in this process, we also have the use of bacteriophages for this to occur, just like we saw with the lysogenic cycle. In transduction, the bacterial genetic material is going to be carried across from one bacterial cell to another by a bacterial virus. So that's all that we mean when we talk about transduction, is the transfer of a bacteriophage's DNA from one host cell of a bacterial organism to another host cell of a bacterial organism, allowing those bacterial cells to exchange genes. This is different from lysogenic conversion because the bacteria are acquiring a new genetic information from a viral gene. So that's our process of transduction. Now, the process of transformation is a little bit easier and less complex because it doesn't deal with bacteriophages. It just deals with naked DNA that could be pretty much all out in the environment. That naked DNA is going to basically find a recipient cell, and it's going to penetrate through the cell membrane. Once it penetrates through the cell membrane, the DNA of that the DNA of the recipient cell is going to basically uptake the new DNA that's been added. So here you see these strands that are identified as A and B, capital A, capital B, and then you have this bacterial chromosome, which is B, A, C, D, E. Well, what's happening here is once we add DNA fragments from a donor cell into this cell, uptake occurs, and then components like B are going to be integrated into the genetic code. So notice how A is not being integrated, it's just segment B that integrates in. So it doesn't have to be, it could just be, you know, a particular segment of the fragmented DNA that is going to be used to transform the cell. The rest of the DNA that doesn't recombine with the host cell DNA is going to be degraded and destroyed. So that's not going to be used by the cell. So this is an example of how transformation occurs. One example is that you can do with bacteria to enable it to express a new trait is you could take a gene from a jellyfish known as P-Glow, and you can take fragments of that gene and put it in put it outside of a bacterial cell like E. coli. And E. coli would uptake the DNA fragments, and then after 48 hours, those fragments would be integrated into the E. coli's chromosome. After integration, then replication of the chromosome, transcription of the genes, and then translation takes place. And so then the bacterial cell will start to produce the gene that originally came from a jellyfish. And this will enable the bacteria to glow or express bioluminescence when exposed to UV light, which is an interesting experiment to see done if you have never seen it done before. Okay, so another way that bacteria are able to get new genetic information is through the process of conjugation, which is probably one of the most simplest of these processes to understand. Bacteria use what's called a sex pilus. It's a sex pilus that's made out of some carbohydrates, and it's made out of pillin. And it's used to attach one cell to another. And then it's basically is a tube in which you're going to have the passing or transfer of a plasmid DNA from one cell into the other. So here's where you have a donor cell and a recipient cell. The donor cell is going to attach its sex pilus to the recipient cell, and then the donor cell is going to unleash or allow a plasmid from its DNA to travel into the recipient cell. And one of the key examples that we have of this is the passing of resistance genes from one bacterial cell to another. So a bacterial cell that receives something that's called an R factor, which stands for resistor factor, is going to be transformed into a cell that did not originally resist an antibiotic that now will resist an antibiotic because it has received the R factor. And this is how we're able to get things like superbugs that can cause a lot of complications. Here's a diagram outlining that bacterial conjugation process. You have here an HFR cell. It's just the name of the cell that has the genes that will allow the production of a sex pilus to exist, and the R factor is going to be included in this. So you have an HFR cell. It will start to express the conjugation tube or the sex pilus, and it's going to affix that sex pilus to a normal cell. So here's our normal cell on this side. Once they're joined together, you have the transferring or the copying of this HRF chromosome, and once this chromosome is copied and it passes through the sex pilus, it's going to make its way to the normal cell's DNA for integration. And once we have integration, you have the loss of the sex pilus. That will start to break down, and then this particular cell here can easily become a new resistant cell because it now has received the R factor. And here is a nice microscopic image of two bacterial cells undergoing the process of conjugation, where you have essentially a donor cell and a recipient cell of E. coli transferring plasmids to one another. So these are interesting ways in which genes are transferred from organisms, especially microorganisms like bacteria. So we talked about transduction, transformation, conjugation, and then the lysogeny, conformation. So those are 4 processes that are essential to the transfer of new genetic material into bacterial cells. So now let's kind of move into talking about the process of genetic engineering, because understanding those 4 processes that we just talked about, it can be very, very influential in the scientific world. Genetic engineering is nothing more than taking DNA from a foreign organism and integrating it into a new organism or a new system. And when the DNA is taken out by that new system, it's referred to as recombinant DNA. So take something like a frog and a turtle. If you took DNA from a frog and you added it to a turtle, this DNA, that would be called recombinant DNA. Or if you've ever seen the movie Jurassic Park, the first one, the best one, there's a couple scenes in there where they talk about, you know, how they were able to complete the genetic code of the dinosaurs. And what they did was they took DNA from frogs and they integrated it in with the dinosaur DNA. So that's what we talk about when we talk about recombinant DNA. We're just talking about adding genes from an original host source to a new host source, which are typically different species. So why is this important to us? Well, this is important to us because it's great for manipulation in the laboratory. It enables us to understand how certain genes and attributes are expressed that are found, you know, in humans, in particular, as well as, you know, other animals. And because there's a lot of genes that are mixed in and associated with certain diseases. So us understanding and using genetic engineering to understand those processes is very, very important. But plasmids, because they're very small circular components and they code for specific genes, they're very effective in being used to be inserted into cells to transfer specific genes, because there's so many genes out there that exist inside of linear DNA, and we don't know what all those genes code for. But with plasmids, because they're smaller and less complex, a lot of times they're more controlled and are better used as vehicles to be inserted into cells.